got that phospholipid bilayer in here. Because as they're leaving, they're taking that with them. So it's got so envelopes will have phospholipid bilayer, they'll have some proteins, they'll have whatever is in the cellular membrane because that's where they made it out of. They just took some of that with them.
So no virus exists kind of like naturally within the envelope. It's always after they've infected something.
I think so.
Okay.
Well, I don't know that we've ever found a virus that hasn't infected something, so that's a chicken egg question.
Okay.
But I don't know that we know of any enveloped virus that really makes its own. It's so easy to steal a host, so why bother? They might make modifications to it, or things to stick in it, but why bother? Okay. Bacteriophage, T4. So talk about this. Head has tail, base, tail fibers, likes to inject. It's a bacteriophage. What is it going to be injecting? What is it going to be infecting?
Bacteria.
Bacteria. So, genome-wise, it's double-stranded, lambda bacteriophage. It's lambda, double-stranded, less than 50 kb, and so it's like you can start seeing just. I mean, it's kind of fun to actually have real pictures of what these look like. For so long, in science, we were going off of cartoons or what we thought they looked like. Now, microscopes are catching up to where we're actually able to see these. I mean, for the longest time, we weren't getting pictures of. We can get a picture of one right now. You know, so these are newer things that are coming along. Is we can know exactly what they look like versus just kind of what we're guessing they look like. Okay. So, bacteria being attacked by a bacteriophage. And we talked about the steps in bacteriophage replication. So there are 6 basic steps. Right? You have attachment. The virus needs to find its host. Needs to enter, get its DNA or its nucleic acids into the host. It a lot of times will then just degrade the host DNA, so that way only its nucleic acid remains. And then it'll take over. So all of the host components are still there. It does synthesis, and it starts making phage proteins. So you get that the copying of the DNA, if it is, the copying of RNA, and then the addition of making the proteins. Then you got to start assembling. And so for some of these bacteriophages, there's a lot of little parts. I mean, you can see you have the base, tail, sheath, DNA, capsid. You know, put it all together, you get the mature head. And then once you have the head piece, you have to put the tail, tail pieces on, the tail fibers. So you can finally get a mature ear.
What within the cell actually assembles these parts once they're made?
Proteins. Because
So the virus actually codes for making proteins to assemble the parts once they're made?
Mm-hmm. The virus codes for everything it needs to make more of itself. And so, basically, you get rid of the host's instructions, so now you only have the phage's instructions. So, the host still, there's all of the machinery, though, to do DNA replication, to do RNA transcription, and to do translation. So, it just hijacks all of, it's basically coming into a working factory and then just saying, everything you're doing, stop, do this now, copy this. And everything's there and working, and the cell doesn't know that it's not following the right instructions. It just knows, I have DNA, I should replicate this. I have messenger RNA, I should make proteins.
How efficient and accurate are they at doing that? Or does it ever mix in with the host DNA?
Different viruses will actually mix in with the host DNA. That's when we'll talk about different cycles. I mean, this is the lytic cycle. The entire point is get in, take over, and produce more and get out. But like HIV, sticks around for a while. So when you're not lysing the cells, then you have to figure out how to stay in there for a while. But they're pretty good at, I mean, they're very compact. They don't have introns. They don't have extra information. So everything they have is key to making more of them. That makes sense? They can make mistakes on their own replication. You know, that's where the retroviruses are horrible, because if it's an RNA code, going from RNA to DNA, they make mistakes. And so, but that still doesn't necessarily affect them downstream, because they still make a virus. Okay, so synthesis, make everything, assemble, put it all together. Once you have a bunch of little, you know, mature virions, stage of babies in there, you break the cell open and burst out. Alien movie. Okay, so lytic cycle. So for the virion, it enters, you start having this downtime where, you know, because it has to kind of disassemble itself in a way so that it can get that DNA out of itself and start being replicated. So that's why you have, you know, if you look on the left side, the number of infected virions in the medium. So it's like, you know, you start with one and then it drops because it's no longer infectious because it's already infected something. And then you have this time where it's trying to synthesize and assemble, and then it lyses the cells. And so, I mean, if you look at this, we're talking, what, 30 minutes from entry to cells dead? So, and then also we're talking about going from 1 to 100,000, however many can fit in there and they can get in that time. So there's a lot of virions being produced at one time, not one cell, which then all of them can go and infect more cells all over again. Questions?
I have a kind of a broader question. So like for colds and other things, disease caused by virus, so how do we overcome that, or how do you know, like what happens?
I believe that goes more for the immune system and ways of fighting, because a lot of times also when, I feel like with us, if we have something that's not working right, the outside cell wall will start having different protein markers on them, basically signaling, hey, this isn't right. And so, I mean, because the way I understood our immune system was the white blood cells are kind of cruising around, tasting the different cells, going, oh, you're yourself, you're not quite right. And so they start figuring out how to attack those cells that have been infected. So if you take out the cell before it has time to let the virus replicate, you keep doing that, then you can do that. And that's a lot of times why we get that fever, because you can kill proteins. So you can start.
So it's like the virus. So what happens is like the viral DNA or RNA. It's just like floating around, and like once you recover, I mean, it's still there.
It'll probably
it's
be
just not
degraded.
in a form it can attach to cells. Or?
Well, I mean, because if you lyse a cell before letting the virus be packaged,
Yeah.
DNA or RNA is not going to survive very well.
Oh, okay.
And so it can get degraded. So having free-floating nucleotides in your system isn't really an issue.
Oh, okay. I see.
So.
You mentioned that as the DNA or RNA within the virus is making copies of itself, it makes tons of mistakes and mutates. Wouldn't that
Not all.
Okay. Those that are doing that, wouldn't that tend to change them into something which might not be pathogenic to humans?
Potentially, but that doesn't seem to be the case for HIV. I mean,
It
HIV
could.
is one of the greatest examples because, I mean, within one patient, they can have hundreds of different versions of HIV. But maybe 1 or 2 of those aren't pathogenic, but those other 98 are still going to kill you.
That what?
So, I mean, enough of it is still preserved. I mean, it changes enough that you can't necessarily make like antibodies or things against it because it just tweaked it slightly enough that you can't, you know, you have a hard time.
Also, once you've had a virus, either a cold or the flu or whatever, are you not Are you not immune to that for the rest of your life because you have copies of the antibodies floating around for that particular
You
virus?
should be. There are some viruses our immune system just can't defeat. And our immune system can't defeat HIV. Mainly because HIV takes out our immune system. So, I mean, that's one of the goals of HIV research is how can we restart that immune system. We'll talk about HIV later. It's kind of a special, nasty bug. But, okay? So,
Thank you for going on.
lytic replication. Remember, we got attachment, entry, synthesis, assembly, release. More pretty pictures. Attachment, highly specified interaction between the virus and the host. The bacteriophage lambda binds to a maltose receptor of E. coli. So it's one of those things where they don't just recognize the host, they recognize a particular specific part of that host. Because that's where a lot of times these drugs are trying to target. It's like, if we can change this maltose receptor, does that mean the E. coli will no longer get infected by lambda? And so trying to do things like that and figure out how can we stop these viruses. So, it attaches, finds its host, finds its happy spot, enters. You get to, injection of nucleic acids, biosynthesis, nucleic acids and proteins, all the things we were talking about. Synthesize new copies of the viral genome, synthesize the proteins needed for that viral coat. So remember, he's had a virus coat, but not a sodium blow. Assemble, talk about all these different pieces, construct complete virus particles, viron when it's complete, from the proteins and the nucleic acids. Release, lysine cells. So phages produce lysozyme. And the cell will lyse, releasing the bacterial phages. Lysis is a fancy name for breaking the cell open. You'll hear it a lot in science. Yeah.
Is there a difference between a bacteriophage and a viron?
Viron?
Or is it pretty much the same thing?
Pretty much the same. I mean, for a lot of things. Bacteriophage specifically will do bacteria.
Okay.
Viron is just the complete virus. Viron. So releasing. Okay, so that's the lytic cycle versus the lysogenic cycle. So this is what we were talking about before, the two different cycles. So you can either have that lytic cycle, comes in, enters, destroys, replicates, synthesizes, releases. Or you can have a lysogenic cycle where it comes in, enters that chromosome, becomes part of the genome, and then just kind of hangs out, sits there for a while. Whenever the cell divides, it gets replicated. So now it's in multiple cells. And so then eventually one day some signal will say, hey, get out of there. So most of the time you'll just keep replicating and dividing, so you'll have more and more of these cells that have this virus waiting to go. But one day, trigger, you get induction, all of a sudden this DNA becomes active, starts chopping up that DNA, you go into synthesis, assembly, and release. Okay, so ultimately they go to the lytic cycle to be released from the cell, but they can sit there, be lysogenic for quite a while. I mean, this is why people can be HIV positive but not have AIDS. Okay, does that make sense? I keep using AIDS because it's one of the more common to know viruses. One of the things that's really scary is that you kind of look at how many of these cells does it get to before it pops out. So, I mean, if it was only in one, it starts to come out, be released, your body has a chance because you can recognize something's wrong and you have, 100 to deal with, maybe? But if it's replicated everywhere, then how many can they all come out at once and all of a sudden you're overwhelmed? So this is why some of those are, they're really problems if they're lysogenic. So, they're really problems if they're lytic, but
Does this mean you could never really be cured of AIDS? They'll never come up with something that can actually cure people because it's been encoded into the DNA of almost every cell by say 5 or 10 years?
But they can come up with something to stop it before it got to that
I mean,
point.
because right now, what they're doing with people is they have HIV, so they're lysogenic, but they can give them drugs that reduce the viral load, so that when it does come out, it kind of, they aren't really being able to produce the virus very well, and so they cannot get full-blown AIDS, but they still have technically the virus. Yeah, HIV is tricky. They're trying. There's only been a couple cures. There's one guy who, so I think they found originally there were some streetwalkers who were immune to HIV. And so there's been some little quirks, and then they found, I think the recent news is 2 guys have been cured of HIV or of AIDS but they had to get bone marrow transplants. Because there's a special, there's a special stem cell that someone had, and since they needed to get that, they picked one that also would cure this problem. But that's not a general public. They're never going to do that because it's way too expensive and painful. There's multiple things they're trying. We'll talk about HIV later. I think there's an entire little lecture on that. Okay, so lytic, lysogenic, the 2 cycles make sense? Yeah, good. Inactivates. Any questions? Understand the 2? If I say lytic versus lysogenic, you know what I'm talking about? Mm-hmm. Okay. Virus lifestyle. So we're talking about virulence. So viruses that kill, lyse, the cells they infect. How virulent is this? How much damage is this going to cause? Temperature. So viruses can undergo lysogeny. They can exist as a prophage, as a viral DNA insert in bacterial DNA, with most of their viral genes turned off. And so this is why we try to keep studying. Going, how is it that they can go into the genome, inactivate their genes, and just kind of hang out for a while? And then what is it that, you get replicated? And then sometimes all you need is a temperature shift, and this viruses can then undergo lytic growth versus lysogenic growth. And so some kind of induction, different viruses have different induction factors, but you get that induction, change from lysogenic to lytic, all of a sudden you go from having inactive genes that are just kind of hanging out to start producing viruses. Okay, so animal virus classifications. The viruses get classified based on what is their nucleic acid, what is the strand type, is it single stranded, is it double stranded, is it RNA, is it DNA, what shape. There's very many shapes that these viruses can take. So, okay, it's another form of classification. Also, are they enveloped? Are they non-enveloped? And so these viruses get classified into families. And so you do the families, you have the genus, sometimes, so that's kind of the general, and then you also have your common name. They're not quite as fully branched out as we have for, our classification families. So what are they? You start with kingdom.
Family.
That's families way down. So kingdom, kings play chess, play.
Kingdom, phylum.
Phylum. Kings play chess on fine green cell. Kingdom, phylum, class, order, family, genus, species.
I remember the acronym, but I didn't have enough time.
Okay. So, this is HIV. Just kind of to give you broad, family, genus, common name, human immunodeficiency. With animal virus replication, it gets a little trickier because we have so many different types of cells. It's not like it's just a bacteria that it's trying to infect with a certain type of injury. The attachment, they're highly specified interaction between the virus and the glycoproteins in the host cell membrane. HIV is kind of nasty because it binds to the CD4 receptor of our T lymphocytes. What do T lymphocytes do? Immune system. Really important for your immune system. So, they specifically bind to T lymphocytes. And so if you're taking out a part of your immune system because it's being infected by viruses, you have issues. Okay?
Would you say that it's not the AIDS virus that actually damages you, but the fact that it takes out your immune system and everything floating around here damages you?
HIV is the virus, AIDS is the disease, as far as I've known, and AIDS doesn't kill you. It's usually a secondary infection because you can't fight off a common cold because your immune system's gone. So it basically kicks you and beats you up to the point where anyone can come and push you over.
So the stuff floating around actually wipes you out after the immune system's been damaged by AIDS.
Yeah.
Have they tried anything to like block the interaction between it with something that looks like a virus?
Yeah, so they're doing, since HIV is such a big thing, they have all sorts of different levels that they're trying based on when did you get exposure. And so they try to stop it from recognizing. So if you can't enter the cell, you can't ever get it. They try to stop it from integrating. Because integrase isn't exactly an enzyme we have normally. So if you can destroy reverse transcriptase or integrase, two, enzymes that are specific for the virus, then you can stop the virus. But if it's already integrated, if it's already reverse transcripted, if it's already integrated, then all you can do is try and keep that viral load down. So that's where a lot of people are at, is with that lower viral load. But they found there was one patient, usually when a mother has HIV, they put her on a lot of drugs before she gives birth, so that way her viral load's way, way down. Because otherwise, mixing the blood, baby ends up getting infected. They found one woman who they found out that she was HIV positive, I want to say very close to when she was going to have birth, so they couldn't give her the drugs. So they started the kid on really heavy-duty loads of the HIV viral stuff, and he actually seems to be cured of it. After
He did have it.
he had the viral load, and so they were able to prevent it, keep it, because they went hardcore, harder than they normally would do. I think they started 6 hours after birth, kind of a hardcore virus stuff. I'll find it and bring it in, but yeah, it's one of those things where this is stuff that's in the news within the last year or less. Probably just in 2013, I've been seeing this stuff. So, there's lots of little new things with HIV. You good? Okay. Viral entry. So, you have receptors on the cytoplasmic membrane. We all do. It's just kind of what tells the cell what it is, and so it can, get signals and whatever. And so, the virus, the capsid, can actually come along, and it can find a nice happy place, and it can either do direct penetration, where it just kind of sits there and shoots its nucleic acids into, the cell. So that would be called direct penetration. So an example of this would be like poliovirus, or you can do a membrane fusion, where you have the glycoproteins, so you've already stolen an envelope, and so the cell's going to recognize this. It's kind of like self, so it's going to accept that in. It's going to become part of the cell's lipid bilayer, and it kind of opens up, and all of a sudden that capsid can drop in. Move that capsid, you now have viral genome inside of the cell. Okay, so either find its happy spot, inject the nucleic acid, or that envelope, it can actually just membrane fuse into it, kind of melt in. And if you see how it melts in, you can also see how it'd be very easy to kind of in a way melt out to become these viruses. One of the things to remember is that these viral glycoproteins, which are being recognized, they're something that the virus has done, but they end up staying as part of the cytoplasmic membrane. So they become part of that membrane. Okay, so direct penetration, viron injects genetic material directly into the cell, poliovirus. Membrane fusion, viral envelope and cell membrane fuse to allow the entry of the nucleocapsid. What is a nucleocapsid? Capsid
DNA and
plus nucleic acid. Nucleocapsid. You'll also see those apparently interchangeable a lot. So nucleic acid. So this is where we're talking about measles, the mumps, those viruses. Membrane fusion. You can also do the endocytosis. So the attachment of the virus to a membrane, followed by the host surrounding it and pulling it in. And we talked about endocytosis. So there are times they can come in. So the adenovirus is a naked virus versus the herpes virus, which is an enveloped that can just be endocytosed into the cell. So here's a little picture of the endocytosis. This is an enveloped virus. It recognizes, gets an extra membrane put around it, comes in. So there's two membranes. This is why we have that endosymbiotic theory with the mitochondrial chloroplast. But trapped inside here is your virus, and it a lot of times figures out ways of tricking the little membrane open so it can release. It can kick off its capsid that was protecting it, and now you have viral genome inside the cell. The whole point is viral genome inside the cell. So, synthesis of the proteins and genomes and animal RNA viruses. Okay, so this is just with RNA. So we have three different cases. We have positive single-stranded RNA, negative single-stranded RNA, and double-stranded RNA. And so you can go through, you can get transcription, so you get that complementary strand, and then further transcription of this. So if you transcribe that, you'll get the negative. Transcribe that, you'll get lots and lots of positives. And so then now that's a way to just completely keep copying your nucleic acids. So then, while you're sending off one of those to start making copies of your proteins or start giving a message to making proteins, this can be making more and more copies of your nucleic acids. Okay?
Are viruses the only, I don't want to call them life forms, but the only entities in nature that have double-stranded RNA?
No, we have double-stranded RNA. They're the only ones who might use that as their storehouse for genetic information, for the most part. But no, we have RNA. There's an entire silencing pathway from interfering RNAs. tRNAs, they have hairpins, which makes them double-stranded in parts. So without that, partially double-stranded weird T structure of our tRNAs, we wouldn't be able to make proteins. So, but you could very easily see how this can just hijack regular cellular equipment to make. Come on, it's copying RNA. What copies RNA? Come on, what's the enzyme used usually to go from DNA to RNA? RNA polymerase. Okay? RNA polymerase isn't going to care if its template is RNA or DNA. It's going to make more RNA. Okay? Another version, you have the negative strand, and you can just simply transcribe it, get the positive strand, and then that could make more copies of your negative. You can also get all of your viral proteins now, because you now have the positive strand. Okay, and then go into assembly. So, and then Versus double-stranded, it unwinds. So you have your positive, act as messenger RNA, okay, so you can get all your proteins. Negative strand, it acts as a template. So viral RNA polymerases can make the complementary strands, and you start making it so you can remake that double-stranded DNA. So, or double-stranded RNA in this case, sorry. So I mean, you need that positive strand so that you can make proteins, but you have one, you can very easily get the other. Questions? So, process of budding, so how they leave. So if we're leaving for enveloped viruses, so we're talking about release. So they're going to kill the host, they want to lyse it. You can either do lysis, break open the host and just leave it, or you can do budding formation. If you want to take that membrane with you, there's no point in punching holes in it. Just kind of envelop yourself and keep going. Okay, so budding formation of enveloped, forms in the envelope around the viruses. If you're budding, you're not going to lyse the host. This ends up being more of a persistent infection, so it can just keep shedding more and more virus, because if you don't actually destroy the genome, you just kind of hijack some of the parts, then you can just keep having it make more and more of your virus, and if you're not killing the cell, then you can, over a long term, just keep taking your viral capsid, finding your viral glycoproteins, getting coated in them, start budding, leave, and you end up being an enveloped viron, right? And I mean, this is the cytoplasm of the host, so it's a method of persistent infection. This makes sense? You either burst the cell open to escape, or you bud, and you can do a much more gradual over time. Keep the host alive enough to keep making more of you. So, if we're talking about a DNA virus, most DNA viruses are assembled in the nucleus versus RNA viruses, which are solely in the cytoplasm. I mean, there is no point in the RNA going into the nucleus when it's not going to be DNA. Not all RNA viruses actually go to DNA, because if it's an RNA genome, it just needs to be replicated as RNA. It's only the ones that want to go lysogenic and hang out for a while that need to become DNA to hang out. It's only the retroviruses. So not every RNA virus will use DNA at all. So enveloped viruses tend to do the budding, the naked viruses tend to do the lysis. Which is virus latency. So, how do viruses stay dormant in a cell? It can go in, it can just kind of hang out, kind of maybe be a plasmid, form itself as be a plasmid, or just kind of integrate. And so there's two different methods of just kind of hanging out and waiting for your time, biding your time. Either as a plasmid, viral DNA presents present in the cell as a plasmid, doesn't have adverse effects, so the cell doesn't really notice it. It replicates as a plasmid, so depending on the type of plasmid, cells are used to having a low copy number, like you might only have 2 or 3 copies, but some plasmids are high copy number, and you have hundreds of copies of that inside a cell. So that's a way to just kind of hang out, replicate your genome a couple times, get passed on, and then by the time you're ready to go lytic, you already have a nice base of operation. Or the other method is actual integration into the genome, and just sit there and wait until it's time, until you have your trigger to go lytic. So this can be years of hiding it, may prolong for years with no viral activity. And if it incorporates in, it's permanent. I mean, it can make copies of itself, and it can make more virus, but it doesn't mean that, if you think about it, if you got this latent infection, and if it's an enveloped virus, then it can just start budding, sending things off to be budded. It hasn't destroyed the genome, and if it just keeps making more little viruses, and it keeps budding off, the cell's not going to die, not going to be lysed, and it's there for the rest of the life of the cell. Questions? Okay, so how do we grow viruses? So when we're trying to make our vaccines and stuff, we have to kind of grow a virus so we can figure out what it's doing, so we can try and make things against it, so we can actually, usually you try to give your body either a weakened, a killed, or a part of the virus. So enough that your body recognizes it as foreign, figures out how to fight it, but also can overcome it. So that way when you see the full-fledged version, your body's like, I know what to do. So you don't have that lag period where your body's trying to figure out what to do. A lot of the viruses we use living animals. We can use embryonated eggs. We can use cell cultures. There's a wide range of growing animal viruses for study or trying to find ways of stopping them. So you can culture them in plants, animals, bacteria. One of the nice things with chicken eggs is that they're pretty cheap. They're also one of the largest cells. I mean, come on, it's a single cell. So, you don't have, you also have a nice sterile environment. That shell gives you a nice sterile environment. You aren't going to have microbes be able to get through the shell. So you can kind of grow things in little tiny incubators in a way that are nice and sterile environments that are cheap. So how do they do that? What you can do is you can actually inoculate into embryonated chicken eggs. There are multiple places that you can actually inoculate. If you want to go into the membranes, the embryo, the amniotic, even into just like the yolk sac. So they have a bunch of different places where you could actually inject the virus and see how it's growing, let it grow up, get a large viral load to work with. I know, poor little chicken. Okay, viral assays. One of the common is plaque assay. You mix a virus with cells, spread it on a culture plate. So kind of like what you guys did, maybe make it a little thicker so you have a nice even lawn. And then you start seeing these viral plaques. And so what it is that, it's kind of weird here to see, but this here is just everything that you see that's nice in one shade is your nice bacterial growth. And then all these little dots are where one of those cells had a virus that lysed the cell and then was able to have the virus around it. So all of these are bacterial viral plaques. So they're spots of no growth due to the fact that the bacteria got lysed. Okay, so the clear areas in the culture dish are where the viruses lyse the cells. So it's one of the common ways to be like, does this work? Is this killing this bacteria? Will it affect this bacteria? What's going on? So plaque assay, very basic, very simple. Mad cow disease. Okay, so we start talking about prions. So prions, they're infectious protein particles. So usually they aren't folded right. So something happened to where these proteins are not folded correctly. And so they lack nucleic acids. They replicate by converting similar normal proteins into new prions. So it's one of those things where you have a protein that isn't working properly that can just go along and destroy or convert other proteins into prions. So kind of, they're not fun. They're not fun bacteria or diseases to play with. One of the most famous one is the spongiformia, so mad cow disease, or scrapie in sheep. These involve fatal neurological degeneration. So, and one of the problems is that they're proteins, but normal proteins are destroyed by cooking. These proteins already are degraded or already not functional, so they don't really care. Heat doesn't necessarily work to kill these. Okay. You can destroy them by incineration, but if we wanted to eat cow, we don't really want to incinerate it first. Currently, there's no treatment for prion diseases, which is what makes it so bad. Okay. So, prions, proteinaceous infectious particles, PIP, they're just proteins, no nucleic acids. And it's mad cow diseases because that's where we have found it the most, and it's a neurological degeneration. Part of the problem with why that became so widespread is because they were feeding parts of cows back to cows, and so if one brain is infected, then you're going to keep getting more and more cows infected. They've also had this with, there are some tribes that eat the brains of their elders when they die, because it's thought as a symbol, very culturally to take on their wisdom, but then they were finding that they were having prion disease. So they're trying to tell you, okay, I know it's your culture, but you're really just making yourself all sick. So it's like, how do you, inform people sometimes of the science behind things? Cooking it doesn't work. Cooking does not get rid of the prions because they're just deformed proteins. You have to incinerate. Okay, prions diseases. CJD. They're fatal. You lose motor control, paralysis, dementia. You get croup, shivers. So most of them have similar side effects, mental issues. Epidemic in the foal population with Papua New Guinea. And sometimes they can have long incubation periods, which makes it so that it's even harder to detect. Because if you only have one misshapen protein going around and turning its neighbors into misshapen proteins, it might take a while before you have enough misshapen proteins to start noticing. Because one bad cell is not nearly as bad as hundreds or thousands of cells when you think of how many cells we have in our body. So, how do prions cause disease? They form plaques in brain tissue. And so, these plaques form holes. So the prions consist of a single protein, PRP, found in normal cells, and then PRP can change shape into a pathogenic PRP scrapie. So, change shape. And then the PRP scrapie, or FC, can then go along, find normal PRP cell or proteins, and convert them. That's great. Okay, so it's something that just, we don't know a ton about it, but it's problematic because there's no way to treat it. So what are we looking at when we're talking about this? I always like, it's a nice pretty cartoon of it, but it's the visualization. So if you're able to see, what does, the cellular versus the prion look like? You have these nice alpha helices. Well, all of a sudden, two alpha helices turn into beta sheets. And so, that's structural change. And same basic information, but just because you change that structure, it's now prion, and now can cause entire disease problems. And go along and make more of these into these. Any questions on 13? Okay, so I won't move on to 15. So let's meet back at letter 40. If I get you 15 minutes,